I'm a director of IRCMS at Toshio. We have been studying the blood cell production system, aiming for opening the door to new world in stem cell biology, such as hematopoietic stem cell purification and differentiation, stem cell niche, and cell metabolism.
What my lab has been interested in focusing on right now is to understand how inflammation induced by infection, microbial dysregulation, natural aging influences hematopoiesis through regulation of HSC behavior.
Especially, I'm working for understanding molecular mechanism of how leukemic stem cells initiated and propagated by alterations in transcription factor, epigenetic modifier, and chromosomal anomalies.
We are particularly focusing on how protein translation and protein folding are controlled, because having a proper structure is essential for proteins to obtain intact function. We have previously discovered that hematopoietic stem cells are not good at folding proteins, thereby being disposed to accumulate abnormal proteins, which result in cellular stress, so-called ER stress. So we are therefore investigating what types of special mechanisms control protein translation and folding in hematopoietic stem cells.
Our goal is to understand the mechanism how regulate HSC functions during hematopoietic regeneration. Now, we have tried to understand these mechanisms from the aspect of the metabolic and epigenetic regulation in HSC.
We are studying how these various types of immune cells are produced in our body. I believe that the key of solving this question is understanding higher order chromatin structures.
Our lab is mainly interested in something called developmental morphogenesis, and we use the context of three biological systems to study developmental morphogenesis.
Our study aims to understand how heterogeneous stem cell population in the skin and their niche microenvironment are regulated in physiological and pathological conditions.
My lab is analyzing cellular dynamics in living animals using two-photon microscope. Currently, we are focusing on the dynamics of neural morphology and activity, which is involved in the brain circuit formation.
We are focusing on the cancer research, and research interest is especially tumor microenvironment. I'd like to address the molecular mechanism of the fibrosis and immunosuppressive microenvironment related with a well concept such as cancer stem cells and stem cell niche or cancer metabolism.
Our main aim is to investigate the causal relationship between heart development and adult onset heart diseases like hypertension, ischemic heart disease, and heart failure.
In our laboratory, we focus on molecular mechanisms involving nutrient sensing and regulating metabolism, cell proliferation, and differentiation. Specifically, we are interested in the protein complex consisting of folliculin and FINP.
So I would think that the research environment here in IRCMS offers the frequent exchange between labs, since we have this structure that there's many different labs with different research activity in a very closed space. So I think it benefits everybody that we have the possibility to share the resources and knowledge and also techniques that we can transfer between people from different labs. So this gives everybody the opportunity to have a big development of technical skills and also knowledge. So I think this is very valuable for every researcher at every career stage.
And here in IRCMS, they have the open lab system. So meaning that there is no war between different labs, so that PIs, staffs and students can easily communicate each other. And we also have the on-lab meeting every week, so in which we discuss and get comments on our research, and also we can learn from other people's research. So that is very good chance, I think, for young scientists like me to learn from other people. I think Kumamoto is a lovely place to live. IRCMS is located in the Honjo area, which is close to the center of the city. There is beautiful nature, traditional Japanese buildings, including Kumamoto Castle. And most importantly, Kumamoto's foods are very delicious. I love seafood, sushi, ramen noodles, meat, and other local food in Kumamoto.
Kumamoto City is a convenient city, but it is close to nature such as forests and rivers. So the living environment is very, very good.
You can enjoy to do hiking and camping in Aso Mountain and swimming and fishing in Amakusa and Ashikita Seas. These places are perfectly nice to you and your family.
Tap water in Kumamoto City is natural groundwater from the mountain, filtered over a period of 20 years. This groundwater system is a natural mineral water production system. Thanks to this wonderful water, vegetables, fruit, meat, rice, and fish all taste good.
And I think Kumamoto is very strong in both the developmental biology research, which is my main focus, as well as on stem cell-based research, which is the focus of a lot of other PIs in this institute. And I think you will have an opportunity, a very rare opportunity in Japan, that to a totally English environment, but very internationalized scientific interactions as well as personal life-wise interactions. And I am sure that if you come and take a look, if you have opportunities, or read some papers that are published by PIs from IRCMS, you will be able to get a fuller view of what's happening in IRCMS. So hopefully after the pandemic, we can see you in Kumamoto, in IRCMS, enjoy science, enjoy life in Japan.
I strongly believe that science is one of the ways that can help to make human societies richer and happier. Master or PhD students are more than welcome to join us. We are looking for ambitious, highly motivated students who love science and want to change the world. I think we always need a new blood to do a new science.
I hope many people can enjoy the stay in IRCMS and have a good career in this global environment in Kumamoto. So my message is just that girls and boys be ambitious, aim high. So please join us and see you in Kumamoto.